Okay, let's try this again now. Okay, so y'all had a couple of questions to answer, right? As I do this. So at the end, I'll just throw up another page. Okay, so it was, oh, I'm on the wrong thing. Right, describe the components of urea, that kind of stuff. There we go. See if I can pull it up. And then sections 3 and 4 are really, really short, so it would be easy for us to finish. I think we're going to finish 17 today, so I think we'll be kind of ahead of the game, which like never happens, but we're going to shoot for it. Okay, this one. It gets into genetics and stuff, which y'all take as a separate class. So I'm more worried about 19. 19 is integration of all metabolism. Like I could spend weeks on 19, but okay. So this was our questions, right? Y'all worked on these? Yes, sort of, maybe. Okay. So describe the source of the components of urea. What is urea? Draw urea. 2 nitrogen groups on either side of carbonyl. Okay. So NCO, N, right? That's urea. So what are the 4 different groups that we're going to have to account for? We have the nitrogen, and that'll be like times 2, right? The carbon and the oxygen. So where does the nitrogen come from? One comes from aspartate. Why would you get it from aspartate? Like, what's the upstream? Where did the nitrogen come from before that? But like the original molecule, it comes from amino acids, right? When you're degrading amino acids, you convert it to glutamate, and then from glutamate to aspartate, right? Yes. And then you harvest that backbone, and so you go oxaloacetate, and you get the alpha keto glutarate, right? Yes. Okay, so where else are you getting nitrogens from? From where? Yes, yes, carbonyl phosphate, but where did it come from to get to carbonyl phosphate? Deamination of what? Glutamate. So glutamate and glutamine, right? Glutamate and glutamine. The glutamine is really from biomolecules that are non-amino acids. And then the glutamate is really amino acids. Okay, where do you get the carbon? From bicarb. Where did it come from? Okay, oxygen. Yeah. Pretty good. Okay, so which 2 amino acids serve as primary nitrogen donors? Right, so really how many different things did we list up above for nitrogen source? We listed glutamate, glutamine, and aspartate, right? But we're saying the primary sources are what? Glutamate and glutamine. Okay, so let's talk about glutamine. Glutamine, we said, is a nitrogen in the biomolecules. So how do we transfer it? Right? What happens? You got to think, where are you making this glutamine? Like if it's in all biomolecules, right? Where are all the biomolecules? Everywhere, right? I don't know what I'm doing with my hands today, but they're everywhere, right? So what are our scientific term for everywhere is peripheral tissue. And where does it go from the peripheral tissue? To the liver. And then once it gets to the liver, what do we do with it? We deaminate it, right? Deaminate. And what do we get? We get free NH4 and what? No. Glutamate. Right? Remember, glutamate has only one nitrogen, glutamine has two. So if we release one of the nitrogens from glutamine, we make glutamate. Okay. How about glutamate? We said that was amino acid degradation, right? We said above. Where does this occur? In the what? Most of this is occurring in one place. Most of all of these processes in the liver, just like guess the liver. So in liver, right? And so we go from glutamate, we're going to deaminate it. We're going to get NH4 plus released. Now tell me what you're going to make. Now it's the alpha ketoglutarate. And then in the presence of these two, right now you have two NH4 groups, right? And you have what? CO2, what are you going to make? That's a precursor for urea. Carbamyl phosphate. What's the difference in structure from carbamyl phosphate and urea? You have a phosphate group in the place of one of the nitrogens, right? So then you have an NH2, and then you have, oops, and your negatives, don't forget your negatives. So if you know urea structure, you know carbamyl phosphate. Yeah? Okay. Y'all keeping an eye on the time for me, right? I guess I'm going to erase this so we can see the text. All right. So we've talked lots about degradation, right? What do we now need to talk about? Biosynthesis, right? Now we need to make molecules. Oh, there was another one? Oh, okay. So why isn't aspartate a primary, thank you. Why isn't aspartate a primary donor, right? Can I go in and add another page? Let's do that. Okay. So part C, right? So it's not a donor because why? So when you're talking about the urea cycle, right, you have part of it that occurs in the mitochondria. You have a couple of reactions that are mitochondrial, and you have a couple that are cytosolic, right? So think about where most of these reactions are occurring. Where are they? Where are the majority occurring? I guess that's a bad question, right? Where do we make carbonyl phosphate? Where are we breaking down glutamate, glutamine? Everything that we've talked about so far was in the mitochondria. So all the breakdown of glutamate, glutamine, you're making carbonyl phosphate, right? All that PHOSP phosphate, okay? All that occurs in the mitochondria, right? So what happens with aspartate? It's in the mitochondria, right? Let's see. Let me see if I can go back and find that. Right? This is a simplified version, but I kind of like it. All right. So everything's happening in the mitochondria, right? We're donating all of our nitrogens to carbonyl phosphate, right? What happens with aspartate? What has to happen to aspartate for it to donate a nitrogen? It has to be shipped out. Then it's going to be used to replace the AMP on the intermediate between citrulline and argininosuccinate, right? So it's not a primary donor. It's like a secondary donor because it's not all happening in the mitochondrial matrix. It's happening out here in the cytosol. Yeah.
So it really doesn't have anything to do with like the 2PI.
The 2PI is for, right, if you make pyrophosphate, what is the metabolic goal? Why is it a good thing when you're cleaving ATP in order to make pyrophosphate? Making the inorganic phosphate, making the pyrophosphate is not energetically favorable. But once you make it, the cleavage of the two into individual inorganic phosphates is energetically extremely favorable. So that's what will drive some of the reactions. Yes. So that's all about energy. Okay, good. All right. So now we can talk about biosynthesis, right? Okay, so biosynthesis of our amino acids. So we're going to look at plant and bacteria for 9 of the amino acids, right? Because not all amino acids we can make. There are some that we can't make. What do we call them? Essential. But bacteria, plants, they make them all. So just realize that when we talk about the metabolism, we talk about the creation of the essential amino acids, it's not happening in our bodies. It's only happening in plants and bacteria. Then we'll talk about pathways where we convert intermediates to amino acids. So if you look, there are really all 20 amino acids can be derived from 7 metabolic intermediates from 3 different pathways. What are the 3 pathways? Glycolysis, pentose phosphate, and citrate. So from those 3 pathways, we're going to make everything. So what's going to serve as a carbon skeleton from glycolysis? Right? Remember, we're talking about carbon skeletons here, not nitrogens. We're talking about carbon skeletons. So pyruvate, phosphoenol pyruvate, and then 3-phosphoglycerate. So we get 3 from glycolysis. We're going to get 2 from pentose phosphate pathway. Do y'all remember from pentose phosphate what some of the common intermediates that were shunted off? Do y'all remember that? So it's ribose 5-phosphate. And then, anybody? No, it's not G3P, but erythrose 5P, 4P, not 5P, 4P, excuse me. So it's only 2 from pentose phosphate. And then your last one, citrate cycle, has 2. What are the 2 in citrate cycle that you see over and over and over and over? Oxaloacetate and alpha keto glutarate. So that gives us our 3 pathways and our 7 different intermediates. So it's 2 from each, except glycolysis has 3. Okay, you ready? And I like to kind of, you see how I kind of color coded it? Glycolysis, I'm going to make green. Pentose phosphate, I'm going to make red. Citrate, I'm going to make blue. So I'm going to continue that kind of theme, right? Okay, so here are all of our amino acids. Now, the thing with this is not all the pathways are perfectly labeled. So you got to kind of keep track of what's going on. So let's talk about glycolysis first. Glycolysis, what do you say is green? Glycolysis is green. Okay, so which were the intermediates for glycolysis? 3, we had 3 phosphoglycerate. What else did we have? Phosphoenol pyruvate and pyruvate. Okay, now what? Let's do, what was red? Pentose phosphate. So let's do pentose phosphate. Where's pentose phosphate? What are our 2 intermediates? Ribose 5-phosphate and erythrose 4-phosphate. Okay. And then what did I say? Blue? Okay, blue is citrate. So go down to the bottom. Our citrate are oxaloacetate and alpha keto glutarate. Do you have to memorize this pathway in each individual? No, you don't. No, you don't. I do want you to know the pathways that you're getting these carbon skeletons from. I do want you to know the intermediates. That's really pretty important. But you don't have to know which ones are made from which cycle. I'm not going to get that crazy with you. So remember, we can't do all the amino acids. We can't make them all. It's just plants and bacteria. So why? Right? That's an evolutionary thing that's happened over a really long period of time. Why is it advantageous not to? Less energy, a lot less energy. So I'm going to come back to this table, but I want to skip ahead. I'll come back. So A is an example of a couple of different non-essential, there's not 2 E's, non-essential amino acids, whereas these are examples of essential amino acids. Can you tell me some commonalities and differences? Essential ones require a whole lot more enzymes in order to produce them. Yes. What else?
The non-essential ones are structurally more like simple.
The non-essential ones are structurally simple and are structurally very similar to the intermediates that they got pulled out of other pathways. So if you go and you look at the intermediate structures versus the amino acid structures, you're going to see that they're very, very similar for non-essential amino acids. Whereas look what we're doing here to histidine, to tryptophan, right? There's way more complexity. So they're very different structurally compared to their intermediates. So it's going to require a lot of energy, a lot more substrates in order to make these non-essential amino acids, right? And so as animals, we have evolved to eat things, right? We don't just eat sunlight, we eat things. And so you're going to be getting some of those amino acids if you eat protein, right? So why go through the trouble of making it when we're just going to eat it? It's pointless, right? So that's why those genes were lost. We don't need them. It's a waste.
Is it, since it's not advantageous for us to make them, is it advantageous for plants and bacteria?
They have to make it, right? Plants, where are plants going to get amino acids from? If it's not a Venus flytrap, you know, there are a couple of plants that do eat meat, a couple. But otherwise, all they get to eat is sunlight. They have to make everything else. So they have sunlight and carbon dioxide, and that's where they get their carbon backbones. But everything else they have to make. So they don't have an option, basically, whereas we do. So if you look at the different essential versus non-essential amino acids, right? So our essential ones, where are we getting them from? From our diet, from food, right? There are a couple that are really, really important, and there are some that have like exceptions and things like that. So I want to talk about a few of them. So look at arginine. Do you see how arginine has a little A above it, right? Arginine is actually made in really small amounts, not a whole lot, very small amounts, right? Because we're going to make it from arginosuccinate. So you're going to get some, but it's a very small amount made. So you're going to have to eat to get the quantity that you're going to need for making proteins and all that kind of stuff. Look at our phenylalanine, threonine, tryptophan, right? What do all those have in common? They're all what? They at least have rings. They at least have rings, right? And so they're very complicated. So what you see is the more complicated the structure is, the more likely it is to be essential. But there's an exception to that general rule. Is there something that has a ring that's non-essential? Proline is not necessarily a ring because it comes back to the backbone. Threonine does not have a ring. It's complicated. It's complicated. It doesn't have a ring, but it's complicated. So tyrosine. Tyrosine is metabolized from phenylalanine. So if phenylalanine is on the list as an essential amino acid and you're getting enough phenylalanine, your body can metabolize the phenylalanine to make tyrosine. So we don't actually make tyrosine. We don't make it. It's metabolized from phenylalanine. And we're going to talk about some disorders and things like that that are really important that have to do with tyrosine, because tyrosine is a very, very, very important amino acid. Okay, so we're going to go in and look at general, how it's regulated, and then we're going to look at overviews of some of the pathways of how the amino acids are created. Okay, so we talked about this. All right, feedback regulation. So do you want to make lots of extra amino acids if you don't need them? No, right? So there has to be kind of a general mechanism for if I'm making a lot of amino acids and I need to stop. And there also needs to be some specific regulation, right? Because you don't have a whole 20, but you have a large number of amino acids that you can make. And what if you're only low on 1 or 2, right? You have to be able to control the system as a whole and control the system individually. So if you look at the overall pathway, take for example, threonine, right? If threonine starts to pile up, if I get lots of it, look at that first little arrow. what is it going to go in and inhibit? Its own synthesis, right? Now, is that the only feedback regulation that threonine participates in? No. It also feedback inhibits itself and therefore isoleucine, but itself and methionine, right? Because if I look up here, if I inhibit this pathway, nothing below is going to happen. So threonine can inhibit itself, it can inhibit methionine, and it's also going to inhibit isoleucine just because isoleucine comes after threonine. Then what else are we going to inhibit? All the way at the top, right, you're going to also inhibit lysine. So you see that with isoleucine. Isoleucine is going to inhibit itself, and then it's going to come back up and inhibit threonine and methionine. Right? So you see this kind of very, very complicated regulation, but that's just because you want to maintain the correct amount of each amino acid in the pool that's available for the cell, right? If you start making some weird protein that you really need, well, you want to still make protein, but you're running low on certain amino acids. So you need to be able to inhibit some and activate others. All right. So let's talk about the general pathways. So pyruvate and oxaloacetate. These are two linked pathways. So a linked pathway, what's the definition of a linked pathway? What do you have to have the linked pathway? A shared intermediate. So what's my shared intermediate between these two pathways? Alpha ketobutyrate. Alpha ketobutyrate makes this a shared pathway. So the essential amino acids, kind of hard to tell in here. The essential amino acids are in bold, right? So the enzymes that are required to make them are missing in us. But remember I said we're talking about both plants, animals, and bacteria. So these two pathways are going to be linked. The one on the left, we're taking oxaloacetate. Oxaloacetate is a precursor from what? What pathway? Go up to the top. So from the very beginning, remember we said we had 3 pathways where the intermediates came from, and we made 7 different, it was Krebs, citrate, right? Okay, where did pyruvate come from? So sometimes you're going to see links between the different metabolic pathways, and that's what we see in this example. So if you're inhibiting one pathway, you still have a backup from the others. Do you have to memorize this? Please don't. Please don't. Please don't. Yeah, I would know the essential amino acids. I would absolutely know the essential amino acids. Yes. Yes. Okay. The oxaloacetate derived amino acids, please don't memorize enzymes. Please don't do that. There are so many, right, that we have to go down and abbreviate them because the pathways get so complicated. But when you start out with oxaloacetate, what can you make? You can make aspartate. So is aspartate a essential amino acid? Look at your list. It's okay to go look at your list. So aspartate is an essential. How about asparagine? Aspartate's not, sorry. Aspartate's not. Is asparagine? No. Threonine? So we're starting to get complicated structures. So threonine, yes. How about lysine?
Yes.
Methionine? Also essential. So compare the structures of threonine, lysine, methionine versus aspartate and asparagine. How many enzymes did you need to get to aspartate and asparagine? 1 or 2, right? How many did you need to get to methionine? 1, 2, 3, 4, 5, right? So like 80,000.
So essentially, the more enzymes.
the more energy spent, the less likely we are to make it ourselves. Becomes essential because we lose the genes that encode the enzymes. They are not energetically favorable. That is correct. Okay, pyruvate. Where's pyruvate coming from? Yes. If I am showing you an enzymatic reaction that makes an essential amino acid, got to be plants. Got to be plants. Okay. Yeah. So we can do this. We can do this, but we can't do that. Everything below is plants. Yeah. We could do all of the non-essential amino acids. Okay. So from glycolysis, we have pyruvate. Pyruvate, we can metabolize quite easily to make alanine, right? Pretty simple structure, not too bad. We can go through a couple of more and we can make isoleucine, valine, and leucine. So when you look at these structures, remember when you memorized amino acids in Biochem 1, right? If you're looking at these structures, when you were learning them, leucine is very similar to isoleucine, right? So the ones that have common structures come from common pathways, right? They share intermediates. They come from similar offshoots from glycolysis or, whatever pathway it is.
So valine shares its pathway
With leucine and isoleucine. That's exactly right. So you can look for commonalities in these pathways. All right. The charismate pathway, I want to talk about a little bit more. So sometimes you'll hear it called the charismate pathway. Sometimes you hear it called the schismate pathway. But this is the pathway that's used for all aromatic rings. So are we going to make aromatic rings? No, we are not. Too much energy, right? So in this kind of a reaction, right, you have to make something relatively long to make a ring. So you're going to take two intermediates and we're going to go through a condensation reaction, right? And now we have a long chain. Then we're going to convert that chain to a ring, right? Now we have a ring. Now we're going to, so this is why they call it either the schismate or the charismate because of our two intermediates here. But these two intermediates eventually lead to our different aromatic amino acids. So if we keep going on the pathway, right, we're going to make tryptophan, tyrosine, phenylalanine. Do you remember at the very beginning of the chapter, we talked about the pesticide, right? So what happens in these genetically modified organisms, right? We have a gene from bacteria. That's the plant synthetase enzyme from right here, okay? So if I put this drug, this herbicide, on my plants, all plants that require any of these enzymes, any of these aromatic enzymes is going to die, right? But if I replace this gene with a gene that is resistant to the herbicide, now my genetically modified organisms can survive because this inhibitor doesn't have any effect. But how about the natural plant life? They die, right? So you have to be very careful of where you're spreading this herbicide. Because if you spread it outside of your cropped plot, you're going to affect the plant ecosystem there, right? How toxic do you think this herbicide is to animals? Why not?
Because we need those.
We can't make these. These are essential amino acids. Do we have the enzymes that this herbicide interacts with? No, we don't have these genes. We can't make it. So how toxic is this herbicide
to animals. It's not. It's not. So are you worried about its effect of frogs in the area? No. Maybe frogs dependent upon the flies who are dependent upon the plants, you know, so on and so on and so on. You might be worried about the food web in general, sure. But directly affecting animals, no. Right?
Just don't have the enzyme for it to.
We don't have the enzyme that the inhibitor affects.
Yeah.
2 minutes, okay. All right, so there are a few things that some amino acids can make that are not amino acids, right? So we can make heme. Where do we have heme? In the blood, hemoglobin, right? Where else? Muscles, myoglobin. Where else? Not us, but well, yes, us. Cytochromes. Cytochromes have that heme group, right? Nucleotides. What are examples of nucleotides? 2 big classes. Purines and pyrimidines. Signaling molecules. Do you remember we said tyrosine was really, really important for making neurotransmitters, for making dopamine, catecholamines? That's right, exactly. So it's really, really important for lots of different things that happen in the body. So porphyrin is an iron containing ring. So if you have heme, you have a porphyrin ring with iron in the middle. So we need it for cytochromes, hemoglobin, myoglobin. That's what we talked about, right? So how does this work? You actually just start with glycine. You add glycine to succinyl-CoA, right? We're going to add it together, right? We have our intermediate. Then we're going to take that and turn it into a cyclic molecule. And then we're going to combine 4 of those together. And now this is the core porphyrin ring, right? This is the very, very beginning of it. And then what we do is we just make some modifications to this main ring structure to make our heme group, right? So y'all know about jaundice in newborns. So when you make heme, your body has to be able to break it down. If your body can't break it down, you're going to have a buildup of the different intermediates, and each intermediate is a different color. So where is heme broken down? Where's heme broken down? Liver. Like half the time the answer is the liver, really. So bilirubin is one of the intermediates that happens in some infants whose liver function is not working quite right. So what do? They put them under a UV light, and the UV light will break down the bilirubin inside of their skin, and they'll turn from that jaundiced yellow to the normal pink baby that you're supposed to see. The same thing is true on a bruise. You get a bruise, right? If you look at the bruise, each intermediate of the bruise will tell you what color it's going to be, right? Brown to yellow, kind of purplish, right? So in the very beginning, it's very purple, right? Because you have lots of heme. So then as heme breaks down, what you're going to see is a transition of color. And then the heme eventually is excreted in feces, and that's why feces is so brown. That's the heme. You always wanted to know that, right?
Sure, we'll say that.
The urobilin is yellow. It's another reason why your urine is yellow, right? So cool. All right, so I'm going to stop there, and I'm going to let y'all come up.
So glad you said that.
Yeah, I try. Okay, so let me stop share. I'm going to leave, and then we're going to, oops, no, share screen. Okay, so I guess I need to log out of my nickel stuff, right?
log out. Okay, so that should clear it and y'all can come in and pull up your presentation.
Take that. Need my pen.
Another password.
Yeah. Yeah. That's how I yeah. Well, y'all know though. Cool. Yeah.
Where is it?
Yeah.
She'll start. She's right.
Yeah.
You might have to log it. It's going to my wrong spot.
Okay, I could try. Let me try.
Yeah, might be my other
Okay, we're gonna.
side. I think it's my other account. But you can- Oh, shit. And you have to do the phone.
Nope, I gotta go grab it. We're so lost. Oh yeah, doesn't it drive you crazy that she set verification?
Yeah.
When you bring your computer somewhere to go and have a meeting, you don't bring your phone, you can't get into it. You cannot. You cannot. IT has fixed it to where you cannot.
I just ignored the two-step verification and then it eventually locked me out. Because when you did it,
Oh, okay.
just my tracer and stuff, so, yeah.
Jaundice. Well, it forces you, it's required so
Right,
much.
yeah. Okay, you ready?
I was just making sure I knew how to
Okay,
change this.
hey, my name's Victoria Briscoe.
I'm Chloe Rowley.
And I'm Taylor Martin. We did our presentation on fatty acid metabolism, fatty liver, no alcohol. So our patient suffers from a fatty liver, but she has no history of alcohol use. So up here, we just have a picture of a healthy liver compared to a fatty liver. So she's coming in for a follow-up of her abnormal liver function test. Our patient is a 43-year-old woman. She returns to her internist after abnormal liver function tests on her routine laboratory evaluation. She has had a weight problem throughout her whole life and has been considered obese since she was a child. She has tried multiple diets to lose weight, but has unable to lose any throughout her life. She has numerous medical problems secondary to her weight. This does include degenerative arthritis in both knees and ankles, hyperlipidemia, so she has a high fat content in her blood, dyspnea, which is troubled or labored breathing in mild levels of exertion, and excessive fatigue. She has no history of diabetes, viral hepatitis, gallbladder disease, or alcohol use. Her current medications are a daily multivitamin and ibuprofen as needed for joint pain. So her family history, she has lots of family history of obesity. She is married with 2 children who are all overweight, including her husband. She has a sedentary lifestyle because she works from home. So this means she does very little to no exercise daily. She has a family history of obesity in both parents and siblings. So her father died at age 61, secondary to diabetes complications. Her mother is 64 years old and suffers from hypertension and coronary artery disease. Her younger brother is 36 years old and also suffers from hypertension. And her sister is 40 years old and has recently started medication for adult onset diabetes. So today for her physical examination, her weight was 248 pounds. This is about 113 kilograms, and her height is 5 feet 2 inches, so 157 centimeters. Her BMI, she has a BMI of 48.5. This is consistent with severe obesity. Her blood pressure was 44/88 millimeters mercury. So her systolic is a little high, but her diastolic is pretty average. Her heart rate was 84 beats per minute. This is average for a woman her age. And her respiratory rate was 16 per minute. She is afebrile, which means she has no fever. Her skin revealed stria, which is stretch marks, with no rash or changes in pigmentation like jaundice. But she does have maceration, moisture, and skin folds under the breast. Her head, eyes, ears, and nose and throat examination revealed full extraocular motion, full visual fields, and normal appearance of the fundus, which is the back of the eye. Her neck was supple and thick without fibromegaly. Her chest examination was remarkable for distant heart sounds with no wheezes or rhonchi. Her heart examination revealed a point of maximal impulse that was difficult to appreciate because of body habitus, and a distant heart sounds with an S4 gallop sound, but no murmurs or rubs. Her abdomen was obese, but with active bowel sounds, and no fluid wave was appreciated. Her liver was not enlarged, but there was tenderness to deep palpation in the right upper quadrant. Her venous varicosities, varicose veins, she has them in both legs, and trace bilateral edema at the ankles. Her neurological examination revealed no focal deficits. So what might be causing the abnormality in her liver function test? This could be due to any damage to liver cells from either infection, immune system abnormality, genetics, cancers and other growths, chronic alcohol abuse, or fat accumulation in the liver. However, we can rule out right away chronic alcohol abuse as damage to the patient's liver because we know she has no history of alcohol use.
So what's normal for a blood pressure for a woman?
I think 120/80.
120/80.
Yes, ma'am.
So when you get to 144/88, you're really getting into stage 2. You're past stage 1. You're getting into stage 2. So you really need to think about doing.
We were a little shocked she wasn't on any medication for high blood pressure.
Yeah, that would definitely be something.
As we can see here, our patient went through a number of extensive laboratory evaluations. First off, she had a normal serum electrolytes, normal blood, urea, nitrogen, and normal creatinine. Okay, thank you. Her fasting blood sugar was 96 milligrams per deciliter. Her complete blood count was remarkable, only for a mild anemia with a hematocrit of 35% and otherwise normal red blood cells. Elevations of serum lipids with a total cholesterol of 238 milligrams per deciliter, which upper limit of normal is 200 milligrams per deciliter. Triglycerides were 452 milligrams per deciliter with a ULN of 190 milligrams per deciliter. Huge difference. Her high density lipoproteins were 49 milligrams per deciliter, and her low density lipoproteins was 158 milligrams per deciliter with a ULN of 160 milligrams per deciliter.
So remember we talked about what's the good, bad cholesterol?
Good is HDL,
Good is
bad is
HDL.
LDL.
Bad is LDL. So they gave an upper limit for the LDL. Why did they give an upper limit for the HDL? Because you want, right, you want a minimum. There is a minimum level. The minimum level, I believe, is about 40 milligrams per deciliter. Right, she was close. Well, so the higher you get, the better it is. So
Correct.
at least she was above that, right? So that's a good thing. That's absolutely a good thing. So that's why, like, when you look at all these things, why did they give HDL as an upper limit? Because there isn't an upper limit, there's a lower limit, right?
As we can see, she has a lot more LDL than HDL levels as well. Her aspartate aminotransferase was 88 international unit over liter, which is upper limit normal of 31. And then ALT was 112, which has the same upper limit of 31 international unit over liters. Her total bilirubin was 1.1 milligrams per deciliter with the upper limit normal of 1.2. And the alkane phosphatase was 148 international unit per liter with the upper limit normal of 120. She had normal blood coagulation tests with negative tests for viral hepatitis A, B, and C, and then normal thyroid function tests, normal serum iron, ferritin, transferrin, and ceruloplasmin. She had unremarkable tests for anti-nuclear antibodies, and then she underwent a CT scan and liver biopsy. Her ultrasound.
Before y'all go on, what do you think from that laboratory evaluation, all of the tests they've done? What is the standout? What are you?
Normal.
Yeah, but like, there are some abnormalities. So what's the big red flag abnormality in that huge long list? Yes, those are high. What else? Cholesterol.
Triglycerides.
Triglycerides. There's your. I mean, like, look at those triglycerides, right? Okay, so these are things that, like, yes, it's a long list, but you need to start pulling out what are red flags, right? So yeah.
Then she underwent a CT scan. An ultrasound examination of the abdomen revealed a normal gallbladder, bile ducts, and pancreas. She had a remarkable CT scan of the abdomen for hypodense regions in the liver consistent with fatty infiltration of the liver. And then the biopsy revealed fatty infiltration of the liver and precellular fibrosis with ballooning and degeneration of hepatocytes and Mallory-Hyland. She was diagnosed with NASH, which stands for non-alcoholic steatohepatitis. I'm so sorry. Histological features associated with NASH are macrovascular fat, inflammation, ballooning, degeneration of hepatocytes, Mallory, Hyland, and hepatocytes necrosis. Although there was no established treatment for NASH, it is recommended that the patient goes on a calorie-restricted diet and increases exercise in an attempt to decrease her weight. Hyperlipidemia was treated with HGM-CoA reductase inhibitors, like we learned earlier, statin drugs, and she started on USDA. Vitamin E was also recommended as it is shown to be helpful. The blood coagulation tests were carried out because the tests are found to be useful to measure blood's ability to clot and how long the blood takes to clot. This can lead to a risk of certain conditions that will block blood flow to important organs. Liver disease is associated with impairment of hemostatic function due to the abnormal and decreased synthesis of the clotting factors. Clotting tests can indicate liver disease, hemophilia, and thromophilia. The causes of non-alcoholic fatty liver disease include obesity, poor diet, lack of exercise, abnormal levels of fat in the blood, resistance to insulin, and type 2 diabetes. Liver biopsy was recommended because it's the only definitive way to provide evidence to detect the severity and diagnose non-alcoholic fatty liver disease. It can be further inferred that our patient has non-alcoholic steatohepatitis because she has a higher level of severity. The USDA is a drug that is beneficial in a number of chronic liver diseases, including NASH. USDA is a particularly hydrophilic bile acid that has the proposed mechanisms of improving the flow of bile as it appears to have hepatoprotective, immunomodulatory, and anoxic properties. The statins reduce cholesterol levels by inhibiting the HDMO-CoA reductase to stop the synthesis of cholesterol and slowing down the production of LDL cholesterol in the liver.
Okay, so in the top left corner, I put a picture of the bagels. Can't really see it, but then we know that that's when we have high carbohydrate, well, non-fat, high carbohydrate foods metabolize to glucose. And then you can see in the bottom right-hand corner, we could see the triacylglycerols get packaged into our VLDLs, and that leads to excess fat storage in our adipose tissue. So I just wanted to kind of keep that big idea in mind. So triacylglycerols are very important as an energy reserve in our body, and our adipose tissue is that repository where we store our triacylglycerols. And for the endocrinology, we have epinephrine or glucagon, which function the same way. What they do is they inhibit acetyl-CoA carboxylase, which we know is the rate limiting, the main regulatory enzyme in fatty acid synthesis. And lipases are going to be upregulated. So lipases are going to cleave to release free fatty acids in our albumin in the serum. And then insulin is going to have the opposite effect of the epinephrine or glucagon. So why do triacylglycerols form vesicles inside cells, and why do they not participate in the formation of cellular membranes? So unlike our glycerophospholipids, our triacylglycerols are completely nonpolar. They have no polar head group, so the hydrophobic effect is not effective. You could say like the entropic effect is not going to be favorable for them to participate in cellular membranes. Instead, it's going to be more entropically favorable for them to be sequestered inside of a VLDL particle. What are the metabolic causes of obesity? So metabolically, this is whenever you're going to input more calories than what you can output. So it's all about that energy charge. When you have more coming in than you can work off, and that could lead to obesity. Additionally, it could also be, they have this thrifty gene hypothesis, which could be genetic, from saying like when we're in a state of eating high foods, like our body would store it from the hunter-gatherer times. And back then it would have been helpful, but now that we're in the industrialized Western world, it's not so much helpful, and it's leading to this epidemic of obesity. And that's the thrifty gene hypothesis. So some results of obesity can be insulin complications, insulin resistance, diabetes, cardiovascular disease, pulmonary disease, reproductive disorders, osteoarthritis, and like Taylor said, our patient had osteoarthritis of the joints, gallstones, cancer, and a serious complication is hepatic steatosis. Steatosis is just fatty infiltration of the liver. So we have the triacylglycerols in our hepatocytes, our liver cells. And this can lead to inflammation and tissue necrosis, death, and that can result in cirrhosis, which is a serious complication. What is the fate of triacylglycerols when blood glucose levels are low? So we know triacylglycerols can be cleaved to form glycerol and 3 fatty acids. Our glycerol is going to feed into our glycolytic pathway. It can be converted by enzymes to our glyceraldehyde 3-phosphate. And then our fatty acids can undergo beta oxidation where we can generate acetyl-CoA, FADH2, and NADH for energy in the electron transport chain. Are insulin and type 2 diabetes related? So there actually is a strong correlation between insulin and type 2 diabetes. And the phenotypic explanation for this is like a higher level of free fatty acids in the serum of the blood. And also peptide hormones are abundant from our adipose tissue. And you can see here we have three. This also kind of goes along with metabolic causes of obesity. We have our three hormones, leptin, insulin, and ghrelin. Leptin is responsible for that feeling of full, like after you eat, whereas ghrelin is going to make you send a signal to your brain to say that you're hungry and you need food. And you can see the expenditure and intake. You want that to be level. Okay, BMI is body mass index. And this is a common way we can kind of measure for obesity. What you do is you take your weight in kilograms and divide that by your height in meters squared, or something more common to us, we would do our weight in pounds over our height in inches squared, and you just multiply by a factor of 703. The normal range is a BMI between 18.50 and 24.99, and our patient had a BMI of 48.5. So medical causes of obesity, we talked about that.
So let me ask something about BMI. Is it accurate for everybody?
No, it's not.
It's really not.
It's commonly used, but it's not an indicator.
It can be really, really misleading. Not to say that, you know, BMI is not something that you can use. You have to be very careful about using BMI as a diagnostic tool. We even calculated our BMI. And we're laughing when it's right. Yeah.
The case study did go into the waist to hip ratio. That could help a little bit more when you take a person's waist circumference divided by the hip circumference. But again, like everyone has different body types. Like you could be pear-shaped, apple-shaped.
For people who are who work out, right? Who have a large muscle mass. I mean,
You have muscle weight, and that can be, it's
and
not.
you have these really, really built people, and like they're obese. And you know they're
And I think more of like a person who's pear-shaped, they're gonna have way bigger hips, but they might not be obese.
So you just have to be careful about using that and, pigeonholing that.
Obesity is defined as excess adipose tissue mass, and we have here that BMI is a widely used index of obesity, and like we talked about, though, it's not a direct measure. I have a picture showing visceral fat and subcutaneous fat. This is important because it can really lead to serious complications, and we could see that the abdominal subcutaneous fats are going to be around our right under our dermis, but our intra-abdominal are going to be surrounding our organs, and that could be dangerous. Our case study mentioned that 22.5% of the US population is obese. I'm not sure when our case study was written, but I did find an article from 2017 to 2018. That's the latest I could find. And it said that 42.4% of the US population is obese. So I thought that was an interesting difference between the percentages, definitely.
Okay, so non-alcoholic steatohepatitis is also known as NASH. It's the most extreme form of non-alcoholic fatty liver disease. It's characterized by hepatic steatosis, which is just fatty infiltration of the liver, inflammation, fibrosis, which is diminished blood flow throughout the liver, necrosis, death of the tissue, Mallory-Hyland, which is just damaged intermediate filaments of the liver cell, or cirrhosis, which is just scarring. And it was first recognized in a group of patients that denied excessive alcohol consumption, but they had liver biopsy results that were similar to those of alcoholic hepatic hepatitis patients.
So why is inflammation bad, right? Isn't inflammation supposed to be a good thing?
A good immune response. Well, non-alcoholic fatty liver disease is kind of in my next slide. It's just the fatty infiltration. That's just steatosis, but then steatohepatitis is the most extreme form. That's when you see inflammation that can lead to cirrhosis, which is scarring of the liver.
Right. So you want to elicit an immune response when you have something foreign going on, right? But when you have something that's not foreign and you're having inflammation, you run the risk of overdoing the system and then causing scar tissue and stuff like that.
So it was first recognized in this group of patients, but they had no alcohol consumption, but their liver biopsy results were similar to that as if they would consume alcohol. And patients were mostly women, mildly obese, and had obesity-related diseases, which is like diabetes or gallstones in the gallbladder. Clinical features were malaise or depression, uneasiness, fatigue, and upper right quadrant tenderness, like that was the location of the liver. NASH is also linked to protein malnutrition, jejunal ileal bypass, prolonged parenteral nutrition, so just other sites other than the mouth to get your nutrition, acute starvation, and medications. And like the lab results showed, she had elevated AST and ALT levels and also alkaline phosphatase levels. And these enzymes are all produced by the hepatocytes in the liver. And so when the liver is damaged, they get released into the bloodstream. That's why you see these elevation in the levels. And so the pathogenesis of NASH is still unclear. It's usually present in cases of steatosis, so just fatty infiltration. A proposed mechanism of pathogenesis just has to do with insulin, because insulin usually suppresses lipolysis. So you don't have as much breakdown of triacylglycerols, but in patients with insulin resistance, then you have an increase in this triacylglycerol breakdown, increase in circulating fatty acids, which can enter the liver, reform TAGs, leading to a fatty liver. So just more fat in the liver, steatosis. Another proposed mechanism is oxidative stress, which we'll see in the next slide. That's what causes the more extreme forms like NASH. It's generated during peroxisomal fatty acid metabolism or by the enzyme cytochrome P450-CYP2E1, which induces lipid peroxidation, and the products of that process is what triggers inflammation, fibrosis, and necrosis. So that's why it's kind of proposed that oxidative stress leads to the more extreme form. Like we're going to see, it's the inflammation in the liver. And also inflammation itself also generates reactive oxygen species. So that's kind of why the pathogenesis is still unclear of exactly why it's caused. So what is the difference between steatosis and steatohepatitis? In this picture, this kind of shows the steatosis is just fat accumulating in the liver. So it's a more benign condition. And then NASH is fat plus inflammation and scarring. So that's a more extreme form, and that can lead to cirrhosis, which is when the scar tissue replaces liver cells. And what are the mechanisms of liver cell injury in NASH? Cell injury in the liver is caused by cell death, inflammation, and fibrosis. And it is proposed that steatosis alone, so just this fatty liver, does not cause liver cell injury, and that the development of a more severe form like NASH requires a second hit to promote that cell injury, and that requires oxidative stress. So the first hit is kind of a hypothesis of the second hit versus first hit. The first hit is just the non-alcoholic fatty liver disease when it may be caused by obesity or insulin resistance. And then the second hit, like oxidative stress, the proposed mechanism producing that reactive oxygen species, increases inflammation, increases cirrhosis, and that's when you end up with NASH, the more extreme form. So that is all. This is our first, we're excited, and that is all.
All right, questions?
What would you do if your patient came in with a history of an eating disorder and possibly could not do a calorie restricted diet? What then would they do? Would that be a big issue?
Well, then that would mean that obesity wasn't the direct cause of the NASH or non-alcoholic fatty liver disease, but you're saying she still does have a fatty liver. But so I guess we'd have to see if she like has alcoholic consumption and then decide if it was non-alcoholic fatty liver disease. But also the high, like, didn't we say that limiting your calories can actually worsen the condition too?
You have to be careful about doing it too fast, right? Yeah. Because if you do that, that, you're actually going to like fuel the cycle rather than it should be a slow calorie restrictive.
We said that like starvation would not be good because your body in the extreme starvation is going to start breaking down your proteins and you need those proteins to synthesize your VLDL. So I think tightly monitor the caloric intake and make sure it's working with that.
So I have questions.
Something like a thyroid cancer, do you play a major role in development of any cancers?
Our patient had no thyroid problems, but it could, I'm sure.
I believe that would be when it starts to be categorized more as Hashimoto's rather than actual non-alcoholic fatty liver specifically.
Curious, because it was like, I've heard people like have problems with it.
Again, with like hypothyroidism, if you have to tightly monitor it, the body doesn't process.
I was just curious because it was,
So what kind of diet would you recommend for somebody with this disease?
Isn't it high protein?
Well, you definitely want low, you don't definitely want low calories, like to monitor the calories. The habitus she talked about, the body, it's gonna be really hard to exercise, you have to monitor what you're putting in, to make sure you're burning it off, but not too drastic.
Liposuction or some kind of weight loss surgery would be beneficial for somebody with this.
I think.
But
that might be a
also,
little risky.
my slide said that like jejunal ileal bypass could lead to NASH. So you gotta be careful, like bypass surgeries might not help it. Maybe liposuction, I'm not sure, but
Yeah, wouldn't it really not help? Because like the environment that the person still was like.
it's,
Yeah.
yeah.
With the liver.
it was just like a
And I think,
temporary
I'm not 100% sure, but I'm pretty sure you have to go through a lot of checks to have that liposuction. I don't know if you're even eligible for.
Yes,
liposuction.
it might help, with weight on joints and things like that, so it might,
The arthritis.
if she was approved for it, it might allow her the opportunity to now become able to exercise more often, you know what I mean, like. But I would think like a bypass, gastric bypass, that would be really, really risky.
It's kind of a two-part question. In the patient summary in the beginning, the patient said she has a family history of obesity. Do you think there's possibly like a hereditary or genetic side of her illness?
I think if the 13 gene hypothesis, like that definitely can be it, like their genes are more activated towards storing fat during times of peace.
And then do you think that her multiple diets may have been worsening and NASH over time? Because she did say that they were unsuccessful, and it didn't say how many diets she had, but it seems like she tried to.
I would say yes, because especially if she just tried to jump on this diet, just
Went
hard,
hard.
no calories. Most of the time, when a person does that, it doesn't end up being successful. I would probably.
But you get the recommendations from every doctor that you visit, you need to lose weight, you need to lose weight, you need to lose weight, so you're getting all this pressure, and then they jump into a diet that isn't beneficial for their
Maybe
body.
a dietitian could be
Right, so
recommended
a
for her to see.
nutritionist, maybe
Yeah.
something like that.
That way they can help her figure out what's a gradual way to do this. Because it's hard to change your lifestyle too, if she,
So y'all touched on the like increasing rate of obesity in America. What do you think the reason, like there's like some reasons are for that, like that is increasing and maybe even accelerating?
the high calorie food, like our industry, and not having the hunter gatherer lifestyle, like, we don't go out.
The sedentary lifestyle.
Sizes in the last like 50 years,
Yeah.
the portion sizes are crazy.
I did hear, like, our small at McDonald's is a large in China. For drink size. I think that's so interesting. It's just so readily available. And we probably eat
way more than what our quality actually is. And I think we cook at home less generally as a population. I think because we're becoming more developed.
Less garden.
Well, less.
Putting ourselves
More processed foods, more fast foods, right? All those kinds of things have a large societal impact. Absolutely.
So like I was looking at cirrhosis and it's like pretty much I was getting it just as like scarring. But like why is scarring so bad for like the liver compared to like your skin? Like you can get like the scar in your hand or
Right.
something, it's not the worst thing. But why is it so like
Well,
I guess detrimental if you get the same like your liver?
the scar would like replace your liver cells, and that's the whole function of the liver. So
And the liver regenerates.
Well, yes, but, so you're gonna have some regeneration, but at some point, when you get to cirrhosis, you're not making new functional liver cells.
Right.
You gotta think about what are the functions of the liver? Like, how many times in lecture today did we say the liver, the liver, the liver? Right?
Metabolic hub of the body.
Oh, the metabolic hub of the body, that's right. So,
It's full of scar tissue.
so how's that different from the skin?
the skin regenerates all the time, though. And
Well,
then
yeah, but like,
It's almost
so
like
like,
it's just not.
Yeah.
it's
It's
not-
just
it's a barrier, right?
Yeah.
It doesn't have as many different functions. It doesn't have as complex of functions, right? Whereas here, in the liver.
Right.
Food store, if you like, if you're losing all this space to store all this food in all this like.
Well, not
this
just
food in the body.
What else we got?
I think maritime.
All right, so the part of the treatment plan was ursodeoxycholic acid, which is sourced from Chinese black bears. So it's one
Yeah.
of those like kind of iffy medicines because it's harvested in a very unethical way and puts the animal in danger. So did you come across any other alternative medication, alternative drug that could be used in its place?
well, that was the one that like directly affected cholesterol synthesis, right?
That
Yeah.
no other exact drug. No. I'm sure there might be more.
Drug development is a huge industry, huge. Ernest, did you find one?
Not anything that would be just as effective because it's kind of a miracle drug.
I don't know if the bears have to be, I don't know the process of it. Yeah, we did, we just saw Chinese black bear.
And we were like, that's insane.
Go out. Maybe a way to get around that is to, can be synthetically created.
Right.
So,
Anybody
That
else?
was a question.
Yeah, me too.
Right here. Oh, but I just wanted to make the point that like, she's not on any medication.
Right.
But ibuprofen.
Right.
So taking that as alternate probably does like just too much ibuprofen. Oh, it's the last.
That's true. That's true. Yeah.
Ibuprofen can also be one of the reasons why the blood pressure was so high.
Yeah.
And then
just recommendation, so, yeah, those research to imply that the patient may be using steroids. Let's just say the patient was using steroids, that have a effect on the liver. And was development. Yeah, so let's just say, that's the steroids, prednisone.
like causing inflammation.
Yeah, the use of steroids.
Yes, but we didn't see anything about it. But I mean, any steroids, it's not great. But
Overuse of steroids can't, but yeah. As needed, very, like, not rarely, but like it's as a by treatment for treatment only kind of issue, they can be.
Yeah, the general is if they ask you to get a steroid shot, they ask you, have you had a steroid shot in the last like 3 months?
Yeah.
And if you say yes, they won't have it.
Right, like you have to have like so much time between, so if you're just using it for a treatment and then like a while later, you have to use it again for like a treatment, and it'll be fine. It's not really gonna have that big of an effect until you can use it on a regular basis.
I don't know if it would directly lead to NASH, but I mean, I'm sure it will have some.
Or yeah, it's in the liver.
Maybe like in her particular You logging out?
Yeah.
Okay, don't forget to just remove our email.
Answers, your little, your questions and answers. We went over it. Checking your time.
I'll check.
Yeah.
And that's why, that's my question.
No, I think I'm.
You don't have a good.
I have to just tell you real quick. Sorry, I'll tell you real quick. I have a huge schedule update.
Okay.
I ended up not doing virology. I ended up doing molecular and cell bio. I reached out to Mallory Robichaud, who said the three most important classes were anatomy, physiology, histology, and immunology.